Hello and greetings from the university town of Göttingen, located in the middle of Germany, and there from the X-Lab, the Göttingen Experimental Laboratory for Young People. In this colorful building on the North Campus of the Georg August University, we receive groups of students from all over Germany, but also from many other countries. We are Erhard Irmer, a chemistry teacher at the Otto Hahn Gymnasium in Göttingen and responsible at the X-Lab for integrating student teachers into our lab work, and Vivian Keune, a student teacher in chemistry. Vivian worked out the experiments on this topic during his bachelor's thesis. In the following presentation, we would like to show you how the photochemical degradation of an everyday substance, riboflavin from vanilla custard powder, can be used to repeat, apply, consolidate and expand important contents of school chemistry classes. For this purpose, we present 3 experiments on the detection and photodegradation of riboflavin. Light and color have always fascinated people, and especially the cold glow, when excited by ultraviolet light, never ceases to amaze schoolchildren. Such fluorescence phenomena occur in everyday life, for example in neon minigolf, but numerous natural substances also show fluorescence. It is even more astonishing that in school chemistry lessons, energy is mainly considered in the form of heat or electrical energy, but light is hardly ever mentioned. Only in the treatment of dyes, the interaction of light and matter is considered. Eventually, also in the formation of radicals or photocatalytic processes. There are simple ways of didactically reducing the key concept of ground state and excited state. In fluorescence, electrons are raised to a higher unoccupied energy level by excitation with ultraviolet light. During the process of emission, these electrons give off part of their energy in the form of heat, that is through molecular oscillations. The other part of the emission is decisive. Here, the energy is emitted in the form of visible light. We observe this light emission as fluorescent light. In our talk, we focus on a representative of the flavins with everyday significance. Riboflavin, also called vitamin B2, is found in various foods, for example in vanilla custard powder, where it, together with other natural dyes, provides the yellow color. Due to its extended double binding system, a riboflavin solution shows a yellow fluorescence with an emission maximum at about 520 nanometers.
In the first experiment, this yellow fluorescent color can be used to detect riboflavin. In addition, riboflavin can also be detected by the addition of silver nitrate by means of an intensive red coloration and the resulting fluorescence quenching. Riboflavin is not stable against light and is degraded to lumichrome upon exposure. In contrast to riboflavin, lumichrome shows a blue fluorescence, as you can see in the picture. Thin layer chromatography is used to better distinguish the colors. A few drops of the solutions are applied to a cellulose plate, which is placed in the TLC chamber. The yellowant is a water-acetone mixture, which can be easily prepared in a school lab and used in a student experiment. Under UV light, the upper yellow fluorescent riboflavin spots and the blue lumichrome spots underneath become visible. In another experiment, various influences on the photodegradation of riboflavin are investigated. For this purpose, the irradiation is carried out at different temperatures and light colors. Also, different solvents are added to the riboflavin solution. It can be seen clearly that at higher temperature and shorter wavelength of light, the photodegradation is accelerated. If we look at the influence of the solvent, especially the addition of ethanol or acetone accelerates the reaction. When using water or methanol, a large amount of riboflavin is still visible on the TLC plate. The photodegradation is slowed down by these strong polar solvents.
At the end of our presentation, we would like to address the question of how this topic can be incorporated into school chemistry lessons. When integrating new, innovative contents into existing curricular structures, they are often shifted into a niche. A link with contents that have to be covered compulsorily often does not take place, or rather arbitrarily. We would like to present a simple and memorable model that can be used to clarify the functions of such content that is not yet at the center of the curriculum. The mnemonic RACE stands for repetition, application, consolidation, and extension. The experiments on the fluorescence of riboflavin are suitable for repeating the atomic structure with the electron shell and energy level model, the principle of absorption and emission, as well as for repeating thin layer chromatography as a separation method and the solvents and their properties. The topic can be applied, for example, to the light resistance of food or medicines, but also to the treatment of photosynthesis. The treatment of mesmerism or delocalization is suitable for consolidation. Solvatochromism can also be an in-depth aspect. Finally, the topic can be extended with photocatalysis or electrochemical aspects. The experiments are suitable for different age levels and offer a variety of approaches for subject connection with biology or physics lessons. In a science camp at XLab in 2019, the topic provided that it can be very motivating and is very well suited for explorative learning. We hope that we have been able to arouse your interest in this exciting and motivating topic. We would like to thank you very much for your attention, and we are looking forward to receiving your suggestions or inquiries. Please feel free to write down your comments or send us an email. Thank you and goodbye.